All right, good afternoon, everybody. I want to thank you all for attending our Diversity and STEM Scholars Series as part of our Howard Hughes Medical Institute Inclusive Excellence Series. So I especially want to thank our speaker today. But first, just a couple of announcements. We're going to disable the chat during most of the talk. If you have a question that you would like to ask our speaker, please use the Q&A function and we will moderate those questions at the end. And then secondly, there will be a student meeting with our speaker today later on, and we will post a link for that meeting in the chat about 15 minutes before we finish. So with that, I would like to introduce our speaker today, Dr. Jelani Zarif. Dr. Zarif is an assistant professor of oncology at the Johns Hopkins School of Medicine and the Sidney Kimmel Comprehensive Cancer Center, and a member of the Bloomberg Kimmel Institute of Cancer Immunotherapy. His research focuses principally on prostate cancer immunology and identifying the molecular mechanisms by which host immune cells contribute to prostate cancer metastasis. As a Chicago native, Dr. Zarif earned both his bachelor's and master's of science degrees from Jackson State University, and then he completed his master's thesis in the lab of Dr. Stephen Akunwe, while a part of the NSF Bridges to the Doctorate program. Dr. Zarif received his PhD in cell and molecular biology from Michigan State University in the lab of Dr. Sidney Morante in 2014. His dissertation research focused on androgen receptor signaling in prostate cancer and elucidated how androgen receptor controls prostate tumor cell adhesion and invasion. He then completed 2 postdoctoral fellowships, one in the Department of Urology in the lab of Dr. Kenneth Pienta, and in oncology and immunology and hematopoiesis in the laboratory of Dr. Drew Pardo at the Johns Hopkins University School of Medicine, both of which focused on prostate cancer immunology and immunotherapy. He has been the recipient of several awards, including the Prostate Cancer Foundation's Young Investigator Award, the Patrick C. Walsh Prostate Cancer Research Fund, and the NIH and National Cancer Institute K22 Career Transition Award. So please help me welcome Dr. Jelani Zarif.
Thank you, Dr. Johnson, for that kind introduction, and welcome to today's lecture. I'm going to talk first about my personal journey into a career in prostate cancer research, and then actually what we're researching a little bit. And my journey into cancer research and where it started and how it's continuing with what I'm doing and my lab is doing today. So here are some of the early years into just science, really. I come from very humble beginnings, so I'll start there. So my grandfather and grandmother got married in Mississippi in the 1950s, and they left Mississippi due to the murder of Emmett Till and other civil rights events that were happening in that time. They were a part of the second wave of the Great Migration to Chicago, Illinois. That's where my story begins. And my grandfather, he was a Korean War veteran. He was decorated. He was one of the best shooters in his platoon, even though it was segregated. And he had an associate's, what would today be an associate's degree in TV repair. So he was trained to do that. And in Mississippi, he was able to, have a small business and do this after he came back home from the Korean War. But when he went to Chicago with my grandmother, he had a hard time securing a loan and also starting his business due to a lot of the segregated and discrimination practices in Chicago, which they thought would be different than what they experienced in Mississippi. And so because of that, they had a hard time, finding work and safe neighborhoods and things of that nature. So very humble beginnings. I went to public schools. My public school is still standing at South Loop Elementary. I'm very proud I went there. It's a fine arts school, but they had also a lot of science in the curriculum. And so I was really lucky to attend that school. And while I was there, I participated in annual science fairs nearly every year in elementary school, as well as when I went to high school. I was just inherently interested in science. I also participated in a lot of the after-school math and science academies at South Loop School. And because it was a fine arts school, they had a band. So I was in the band as well. So I did recorded band, beginning band, intermediate band, advanced band, etc. I love playing my trumpet. I still do. I think music is probably my first love, with science being a very, 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 very close second. And so after elementary school, or while I was in elementary school, I was approached by a band director from a high school that wasn't far, and he heard our concert band because we won some award, and he wanted to recruit students for his marching band. Now, he was a young band director, fresh college graduate actually, and so he talked to me about going to college. Now, he was the first person to actually talk to me about attending college. At this time, there was a television show called A Different World that was on TV, and it highlighted historically Black colleges. And I was watching the historically Black college bands at that time too. And so I had this band director who went to one. He was from the local high school, and he actually brought his high school band up to our school, and they performed. And it was like seeing Drumline for the first time and never being aware of it. And he told me I was a really good trumpet player, and if I attended his school and was a part of his band, he could help me get into college. So that was it for me. So I went to a high school named after Paul Laurence Dunbar, a poet in Chicago. I attended this high school with the one and only Jennifer Hudson. She was 2 years my senior, and she was in the choir. She was a music major. And when I was there, you could choose a vocational major, and she was a music major. And I chose medical technology because I wanted to become a physician or a scientist. Of course, with this recruitment from this band director named Dr. Koger, I was a part of the concert band, brass wind ensemble, and the marching band. So we played football games, and it was a lot of fun. I was part of like the spirit of the campus, so to speak, when I was in high school. And while I was in high school, there was a local community college called Dawson. It's still there in Chicago. And they partnered with local high schools to recruit some of their smartest students who were interested in careers in the health sciences and healthcare fields while we were in high school, to do a licensed practical nurse certification, which was 2 years, or a certified nursing assistant certification, which was an 8-month program. I was already a senior, so I couldn't do 2 years of the LPN, which is what would have been my choice. So I was a rising senior, like they presented this to us. I was completing my junior year. And so I did the CNA program. You had to test to get in, and several of the students in my medical technology shop is what we called it. We graduated as certified nursing assistants from high school. And so as a high school senior, I worked at a nursing home called Chevy Chase Nursing Home. That's also still standing. It's called Renaissance now. As a senior, it was, before I became a professor, the best job I ever had for lots of reasons. I got to work with doctors, I got to work with nurses, I got to take care of people, I got to talk to people that could be my grandparents and people that were older than my grandparents. I even took care of a centenarian. She was 101, and she marched with Dr. King. So I got a lot of inspiring stories from working at this nursing home, but I took care of patients who that were somewhat independent on my floor at Chevy Chase. Some had cancer and were recovering. And so that led to my interest in cancer research and going into a major where I could either become a physician or a scientist. So just a little bit back on high school and my college transition. So I'm not literally tooting my own horn here, but I was a pretty good trumpet player in high school. I was section leader, I was first chair. A lot of colleges recruited me to go and play in their bands in college. But I ended up wanting to go to Jackson State University for several reasons. For one, I earned a scholarship there, a partial scholarship, because I wasn't a music major. I didn't want to major in music. I didn't want to become a band director or choir director or any of that. I knew that pretty early on that I wanted to go into the sciences. Even though I loved playing my trumpet, it was more of a hobby. I couldn't see myself at that time having this as a full-time job, because the arts are really, really tough and competitive, as some of you may know if you've played in the instruments. So I tried out for Jackson State University Sonic Boom Marching Band. It's been rated one of the top marching bands in the country year in, year out. And the other reason I chose to go to Jackson State University, again, I was telling you, I watched the different world growing up, but it was a HBCU. But more than that, it was known for being a research-intensive HBCU and producing scientists. Those are people who earn PhDs, as well as physicians, those who earn MDs or MD PhDs. Lots of them. They had a long track record. And this was before anybody was talking about, diversity and inclusion. This was before 9/11. Jackson State had this history of doing this. So I wanted to go there because of that. And then the chemistry department was ranked in the top 50 chemistry departments in the country because of research publications coming out of the department, and as well as where students went. And it was the only HBCU at that time to be ranked in the top 50. That's 50 period, not 50 HBCUs, just 50 period. And so I wanted to go there. And given my experience as a CNA, because I worked as a CNA for like 9 months, and after I graduated, I went off to college. They let me come back for Christmas breaks to work here and there. But the uninterrupted time that I worked, I knew I wanted to study cancer because it was such a debilitating and disease that affected so many people, whether it was a spouse of a cancer patient or a grandson of someone who died of cancer. They were affected by the demise of their grandfather. And so I wanted to understand how this disease went to other parts of the body and why it caused so much havoc. What was the cause of it? Was it genetic? Was it diet? Was it, was it something that we could prevent? And what's the science behind it? And so I went to Jackson State with those interests. I was 17. And I didn't know a lot about the world. I think I'd only been out of Chicago once or twice. And I come from, public schools, humble beginnings. I had friends that were really good students, and I had some friends that were not, really good students. I had a diversity of friends. I wasn't that type of kid that was, in all the AP classes. I don't think I ever had an AP class I didn't have an AP class in high school. I graduated 29th in my class. I had some B's, I had some C's on my report card, so I wasn't like this super duper straight A student. But I did study hard. I tested well, and I think I did all right, graduating 29 out of 400 something students. So I went off to Jackson State. I actually took the train by myself from Chicago to Jackson. It's like a 16-hour train ride, and that was my second time doing it when I went to college. The first time I went, I had to go down for orientation, and the plane ticket was too expensive for me at that time. So the train was cheaper. So I was like, oh, I could just jump on the train. So I jumped on the train, went down to orientation, got all my classes, got my dorm, because you couldn't do all this online back then. tried out for my band scholarship, all of that during this orientation in July of 2001. And then in August, I went down on the train by myself. Because I came from pretty humble beginnings, I went in pretty focused. I didn't do a lot of stuff that I think was common among undergrads. Yeah, I went down because I come from humble beginnings. I said, I have to make the best of this opportunity. I didn't do any drinking. I didn't smoke anything. Never have. So I wasn't into all of that. I didn't need to go to. I went to parties here and there, but I didn't go to all the parties. I was pretty focused. I remember being in the library a whole lot in undergrad. But when I got there to Jackson State, I was pretty encouraged by professors that I had and the upperclassmen that I interacted with. I was just so inspired. I could still name a lot of them who were aspiring to go to medical school or get a PhD. When I was in high school, there were smart kids, but none of them really wanted to be like physicians and things like that. They wanted to go into accounting and this and that and nursing, but no one ever talked about the PhD at my high school. I will say being in a marching band of like 300 people, band practices were pretty long. So I had to learn how to effectively study as a biology pre-med major and be at band practice. And so I would study before and after practice. So there are a lot of nights I would just take my instrument and go right to the library. It was not far from where we practiced. We had indoor practice where we learned all our songs, and then we had to go to the field and do the entire field show for halftime. And we did that field show, I'm gonna say on an average of 7 to 12 times a night. So it had to be like, the lines had to be super straight, the angles, the sound, everything. And we practiced in the rain and in the mud and whatever back then. And so you had to be at practice on time. So I'll get to a lot of why these skills are still important. And I began doing some internships at the encouragement of some of the upperclassmen that I knew, specifically the now known as Dr. Justin Turner. He was an upperclassman who tutored me in chemistry at Jackson State, and he had did an internship at this program. And he said, "Yo, Jelani, it'd be good for you if you did this research internship based on some of your interests." And so he connected me with the program director, and it was at the University of Illinois. And I got to do some research in cardiovascular disease research, just so I could learn how research was done in the lab. I wanted to do research at Jackson State, but most of the labs were already full with students, so there was no opportunity for me, so I had to do an internship. So after a few summer internships, I did 2 at the University of Illinois in the same PI's lab, looking at sarcomere dynamics in congestive heart failure in mice and in some of the isolated left ventricular tissue from those Sprague-Dawley rats and mice. I joined the lab of a research professor named Dr. Stevie Coonway. Dr. Coonway has since passed, so I want to dedicate this seminar to him, he was such an inspiration. He was my genetics professor. I did really well in his class. I think I got an A in his class. I think I scored 100% on the midterm, and I studied really hard. His class was known as being one of the harder classes, and I did really well. And so I told him I wanted to study cancer, and I saw he had a grant looking at this Nigerian basil called Ocimum gratissimum, which is found in Nigeria. It's not native to the United States, but it has a lot of folklore medicine tied to it. So in what I mean by folklore medicine is a lot of people using teas and soups for headaches and sinus congestion and things like that in African countries. In India, they have a similar plant from this family called Ocimum sanctum. It's known as the holy basil, and it has also some other known medicinal properties that go back hundreds and hundreds of years. So his lab was really studying the effects of this plant extract on cancer cell fates. And so he had the plants from Nigeria growing in our greenhouse. We had a greenhouse at Jackson State, and we would go pick the leaves, ground them up, lyophilize them, which is a freeze dry method, and then we would add water to dissolve them into a solution. And then we would measure the solution and milligrams per mil of the solution, and then give certain doses of the extract to our cancer cells. I was working with a master's student at the time. His name was Ron Bozeman. He also went and got his PhD from MD Anderson down there in Houston. And he was working on a prostate cancer project. And so Dr. Ikunwe assigned me to work with Ron on prostate cancer, and that's how I got started. And I've been in prostate cancer ever since. I will say I was really, really well mentored by Dr. Ikunwe. He was a outstanding professor. He knew his science through and through. He cared about his students, so much so that he sent over 25 of us have completed PhDs, and he only worked at Jackson State for 20 years. So nearly every undergrad that worked for Dr. Ikunwe, or every master's student earned a PhD. Only one earned a PhD from Jackson State. The other 24 went to other schools, Nebraska, Columbia, Michigan State, Michigan, MD Anderson, University of Florida, Georgia Tech. Students went to schools after working with Dr. Ikunwe. He was fantastic. And so I did a lot of my undergrad cancer research in his laboratory. I did an undergrad project, independent study project, known as undergrad thesis with him. And then I did my master's thesis in his laboratory. So I want to just talk a little bit about the importance of some of the soft skills I learned in college, and some of which you can incorporate with some of the things you may be doing now in your undergraduate careers. And so I learned this from marching band and from being in martial arts. I did martial arts growing up. Is being on time actually means being early. These are some of the skills that also don't require any talent. So when I was in the marching band, the band director would do the downbeat at 6 o'clock exactly. So that meant at 6 o'clock Central Time, you were already warmed up. You'd already done all your scales. Your horn was not ice cold, or your woodwind instrument was not ice cold right out of the case. You were already warmed up, ready to play at 6 on the dot. Dr. Michael Magruder would drop the hand at 6 exactly. He would look at his watch. So I learned that being on time actually meant to be early. And a lot of that came with like performances. You had to be early for performances, warmed up, stretched, yada, yada, yada. And then in the lab also is being on time. Being coachable. I learned this lots of ways. My karate instructor, my sensei, you had to be coached and taught, I should say. And in any music ensemble, the conductor is going to tell you, hey, you're out of tune, maybe, or you should play the dynamic in this stanza like this versus that, or your attack on a note is inaccurate based on the dynamic that where I'm conducting. So you have to be coachable. Follow your passion. I follow my passion. My passion was cancer research. Your passion may be different from mine, or maybe the same. And don't be afraid to follow what you're passionate about. It doesn't require any talent to do that. Being prepared. So this comes with being coachable and being early. And it's just doing the work, being on time, being warmed up, your horn is not cold, having a positive attitude absolutely goes into everything that you can do in the lab, in science, and if you do music as well, having a positive attitude can affect your playing. Work ethic. In the lab, we don't necessarily practice. We do experiments at least 3 times to make sure they're reproducible and that they're real. We practice techniques so that we can do those approaches. So when it comes time to test a hypothesis, we don't have any bugs in the approach. So that comes with it, and that comes with work ethic too. You have to put in work. I told you I spent a lot of time in the library at Jackson State, studying and putting in the work and doing that. I do think that maybe this requires a little bit of talent, watching, listening, and learning. When I was in the band, you had to listen to other people's parts to know, what was going on with the rest of the piece. When I was in martial arts, you had to watch people's footwork to know if they were about to throw a kick or if they're about to punch you, because you have to learn how to get punched too, as part of doing any contact sport. And learning, that comes with being coachable. There's always something to learn in those spaces. And doing extra, I mean, this can apply to lots of stuff, but I think doing extra, going the extra mile, will enhance all of the other things listed here. So for me, I don't have a picture of me in a dojo or anything. We didn't take selfies like that in the 90s, and I don't remember anybody ever bringing a camera to karate school. But for me, we talk about STEM, but for me, it was STEAM, because the arts were just as important as the science. Like I said, I think music was my first love. Some of the first music I heard was, Stevie Wonder, Michael Jackson, Miles Davis, Herbie Hancock, John Coltrane, Charlie Parker. My family was crazy about them. My mom played the flute, my uncle played the clarinet, and Earth, Wind, and Fire was everywhere. They were from Chicago, so you heard that blaring out of everybody's speakers. So I love the sound of music. But I also love science because, if something, I wanted to know how the radio worked, or why records spun one way and produced the sound, or if the Nintendo was not working or the controller was not working, how can you fix it? What are these screws? What do they lead to? So having that inherent curiosity played into my love for science. But, for sure, and I will say, I'm not just all jazz. And, the early hip hop, like before there was any, bad language in the art and in the music, I really loved LL Cool J, Eric B. and Rakim, a lot of the early pioneers in hip hop. My late uncle David, he used to play the Grandmaster Flash and Furious 5 records. And because there was no bad language, little kids you could listen to it. And we saw older kids in our neighborhoods breakdancing. I had cousins that breakdance. So, the arts and music and the sciences were really critical for me. So here are some pictures of some of my scientific mentors over the years. I do have my mom here, my grandma. They helped foster a lot of my science when I was, doing my science experiments based on books from home for school. So I got my start in science from books that they provided. This is Dr. Ikunwe here. This was my master's commencement from Jackson State University. Did my PhD with Sidney Moranti, shown here. This is us in the lab, me and another grad student, Laura Lamb, who's a professor now at Oakland University in Michigan. This is my postdoctoral mentor, my first postdoctoral mentor, Kim Pienta. I won the Prostate Cancer Foundation Young Investigator Award. They only gave out 26 of them that year internationally, and I was one of the recipients. And so this was at the announcement of the award in Carlsbad in 2016. Yeah. And then this is my last postdoctoral mentor, Dr. Drew Pardoll, who, if you have taken immunology and heard of gamma delta T cells, he is the discoverer of those, as well as some of the known cancer immunotherapy targets in the field. He's in the National Academy of Medicine. and National Academy of Inventors. And both he and Dr. Pardo and Dr. Pinto are both here at Johns Hopkins University School of Medicine. So here are some things that I think, and this is an unexhausted list, of course, that you can do to ensure a career in science. So I think going to class, really admissions committees look at science grades. So making sure that your grades are really good in those science classes, and that's, like I said, doing the extra, shadowing people, whoever interests you for a summer. There are a lot of shadowing opportunities for undergrads, and they pay well. And some of them right now are virtual, so you could probably make a couple grand being home, so you don't have to travel and worry about transmission of the virus. I have always had like a village. There's an old African proverb that it takes a village to raise a child. So I told you about like my band director from high school, my sensei, my family, and teachers at Dunbar High School. They were my village, and they were all positive minded, and they wanted me to, they helped me to excel. And so I think, establishing a village of people in STEM can also be very helpful in finding a mentor. I had several mentors, as I showed in the slides, but in high school, none of my mentors were scientists. Mr. Koger, he didn't have a science degree, and my karate instructor, he had an associate's in psychology. He wasn't a biochemist or a cell biologist, but he told me everything that I did, I needed to, start strong and finish strong, and follow my passion, what I was passionate about, and he encouraged that. And so finding those people can sometimes be a little challenging, but sometimes it can be pretty easy too. So try to find a mentor now, and this mentor doesn't necessarily have to look like you. I know that's maybe counterintuitive, because some of my mentors looked like me in science, but in fact, the majority of my mentors were not African American in science at all, but they helped me excel. So I think you have to find the people that you want to be like, no matter how they look or what their gender is, and see if they'll take you under their wing. And compose a roadmap with your mentor. They'll help you get where you want to go. If you sit down and say, I want to go into this field because of this, or I want to pursue this degree because of that, they will help you if they're a good mentor. Let's go a little bit back to the village. So the village, again, should be positive-minded people. And from this village, you should be able to find at least one mentor, I believe. So this is someone who what you would like to emulate in a few years or be in a few years. And they should be working probably in a similar or exact area you want to work in. Your mentor should be somebody who inspires you. And again, compose a roadmap of what you would like to do in that field as you become an older adult. So I can stop here. I see that there are a few questions in the chat. Or am I reading that? Oh, never mind.
No, there's no questions for you yet. So.
Okay. All right. I thought I saw number 4, and I thought those were questions, but I remember you deactivated the chat. So all of that, it's like under the iceberg, the mentors, the high school program, CNA, these things still motivate me today. And I just wanted to show a few photos of my laboratory. Again, this photo was taken in August of 2019. So before the pandemic reached American shores, so we don't have on masks and all that. But I'm extremely, extremely proud of my team. a lot of people talk about diversity. I just have people in my lab that are passionate about what we do. And with that, you will get diversity naturally. So I think my lab represents 3 or 4 countries. How many languages? 3 or 4 languages. Stan speaks Mandarin and Cantonese. So, but she's an MD from China who came to work with us. Actually, the tallest person in my lab is a high school student who he got a full ride to multiple colleges. He ended up choosing University of North Carolina, Chapel Hill. Debeb, shown here, is an MD PhD student in immunology at Johns Hopkins. So he's doing both his MD and his PhD at the same time. And he defends next month. Rawa is an undergrad at Johns Hopkins. She's a rising senior now. Lauren, she, shown here, she just started medical school last fall at Case Western Reserve. She's a biophysics major here at Johns Hopkins University. And Tom Nerschel is a PhD student in pathobiology. So Tom Nerschel is doing his PhD, Debebe is doing his PhD. And Raekwon, the high school student, he learned how to clone, he learned how to do a lot of biochemistry and molecular biology. He's actually doing some prostate cancer work now at UNC. And Debebe is working on a novel prostate cancer gene that we believe, when altered or knocked out, can inhibit tumors from growing. And Tom is working on a similar gene, but on a different chromosome. So this is my group. We have 2 other grad students who joined last year, who are not pictured, Manali Praharaj and Xiaoshu Wang, who are in the pathobiology and immunology graduate programs. So we study prostate cancer. This is why we do so. It's one of the most commonly diagnosed cancers in American men, with over 160,000 cases each year in the United States. When it's a small tumor, it's usually treatable. However, when it recurs, it doesn't respond to the same treatment. So it develops resistance mechanisms over time due to a host of molecular biology signaling that the cancer cell does. And close to 32,000 deaths a year in the United States. And in cancer immunology, we call tumors cold if they have poor infiltration of cells called T cells. These T cells are part of the adaptive immune system. They can target cancer cells because they recognize them as being foreign, so they can, wipe them out. But when there's no T cells around to wipe them out, the cancer cell continues to grow and grow. And so we call that a cold tumor. If a tumor is a hot tumor, there are a lot of T cells there, and the tumor has really no chance at evading all of those T cells, okay? So this is what prostate cancer looks like when it becomes metastatic. So it metastasizes to the bone. What you see here is a technetium bone scan of prostate cancer from one of Dr. Pienza's patients, where you can see metastases in the rib, shown in black, the spine, the pelvic region, and the patella. You can see those clearly. And this is what kills the patient. It's not localized prostate cancer. It's when it goes to other parts of the body and wreaks havoc. So there are 4 ways of curing cancer that have been proven and FDA approved. The first way is surgery. So you can remove the tumor or remove the prostate gland. The second way is radiation therapy. You zap it with radionuclides. Third way is chemotherapy. The 4th and newest way is cancer immunotherapy. So this therapy uses antibodies to boost your immune response. So your cancer can now be seen by your T cells, and those T cells can go attack it and wipe it out. The caveat with the immunotherapy is that prostate cancer doesn't respond positively to immunotherapy when immunotherapy is used as a single treatment. Other cancers do. Prostate cancer is one that does not. And so these are just some papers from one of the best medical journals in the world, New England Journal of Medicine, from investigators here, Dr. Suzanne Topalian and others that looked at men with metastatic castration-resistant prostate cancer. Again, this is the recurrent prostate cancer that is hard to cure. They tried to treat these men with immunotherapy, again, the fourth pillar of cancer treatment, and none of them had favorable responses to the immunotherapy, meaning that their immune system didn't wake up enough to wipe the tumors out and make the gentleman cancer-free. And this is just another study driving home that point. And so what my lab works on, so most of those therapies are T-cell focused. Those are part of the adaptive immune system. You have another immune system called the innate immune system. It's more non-specific, but it's the first line of defense. And the cells that we work on are called macrophages. The macrophages we've known about since the mid-1800s. A guy named Michnikov discovered them, and he was one of the first recipients of the Nobel Prize ever in science for his discovery of macrophages. Macro meaning big, phage meaning eater. So that's how they got their name. And what we know about them is that they're laid down during a process called ontogeny, during human development, in practically every organ in the body. So in the brain, they're called microglia. So you have thousands of them, millions of them in your brain. In your spleen, you have splenic macrophages. In your liver, you have Kupffer cells, which you may have heard of from your anatomy and physiology classes. And in your lung, you have alveolar macrophages. They all have the same function. If they see something that's foreign or something that may be pathogenic or cause you harm, they will phage it, they will eat it, they will digest it and clear it out of your body. You also have them in your eye. You have them in your bone too. So they're all over your body. What we find is that these cells end up being tricked into helping the tumor grow. And so many labs are now studying this, where these cells are known as being myeloid cells, if I didn't say that. What we find is that other labs are now studying this, and they're looking at the population density of cell types in certain tumors. So we have bladder, breast, bowel, stomach, and lung here. And what you can appreciate from this schematic is the majority of cells that are found in these tumor types are macrophages. They're not gamma delta T cells or CD4 T cells or CD8 T cells. These are the cells. And these macrophages exist in two types. I won't get too into the details just to keep it pretty general. One is tumoricidal. So one can recruit T cells to tumors to help wipe out tumors. The other ones help tumors grow. These are known as M1 and M2 macrophages respectively. And they have a huge subset of a continuum of diversity. We also believe from research that you can convert an M2 macrophage to an M1 macrophage, at least in the in vitro setting, in your laboratory setting. And people are investigating whether this is true in vivo models of cancer. So these are like preclinical models of cancer. So here's a nice cartoon just for for pedagogy, where you have these macrophages have tumor promoting functions, meaning they can cause a tumor cell to go from one and then divide by mitosis into two. So how do they do that? These macrophages can make growth factors such as epithelial growth factor, interleukin-6, and tumor necrosis factor that cause cells to go into mitosis, cancer cells. They can also supply certain proteins and factors for the creation of new blood vessels, which all tumors need so that they can have a supply of nutrients to them. And so this process is called angiogenesis, the formation of blood vessels from pre-existing ones. And so here are some known factors. Macrophages can also make things that can cause tumor cells to invade into other parts of the prostate or potentially other organs and get out of the prostate and go into other organs such as lymph nodes. And these are some of those proteins: matrix metalloproteinases, epithelial growth factor, and cysteine cathepsins. And then they can suppress the immune system. So when T cells come in, they can suppress the immune response that T cells may have. And so these are mostly done by cytokines, which some of you are learning about now in your microbiology and probably immunology and cell biology courses. But cyto meaning cell, kines meaning movement, cytokines like cytokinesis, cytokinetic. These factors cause cells to move or to do something to stop moving. And so they suppress specifically like IL-10 is a cytokine that can tell T cells to stop dividing, stop undergoing mitosis, right? So, and another one that's big here at Hopkins in the immunology field is programmed death ligand 1. So some cells can upregulate this ligand to tell T cells to stop dividing as well. Particularly cancer cells can do that when macrophages make these factors. There are good macrophages in the fight of cancer immunology. Some have anti-tumor function. So when they make specific proteins, they can direct tumor cell killing. And some of these are here, like nitric oxide, reactive oxygen species, which you may be learning about in some of your biology classes, and tumor necrosis factor. And then you have type 1 interferons like interferon gamma, IL-12, and other T cell chemoattractants. So I don't think any science talk is really good without a good hypothesis slide. I certainly know that you won't get a grant funded without a hypothesis being clear and well described. And so this is our lab's ethos and hypothesis based on what we've published and what others have published. So within the prostate gland, there are cells that are epithelial in origin. Prostate cancer is an adenocarcinoma. It arises from epithelial cells. These are cells that are that form the epithelium. These epithelium are part of the prostate gland. And when they come in contact with monocytes, which are precursor cells for macrophages, it causes these cancer cells to transform into a more aggressive form of the prostate cancer. So they undergo a different a genetic shift to a mesenchymal cell type. So that's known as epithelial to mesenchymal transition, EMT. Doesn't stand for emergency technician. And when we put these cells into animals or into specialized wells in the lab, these cells are very invasive. They'll degrade collagen and other basement membranes and go to other parts of the body or other parts of the well, versus these epithelial cells, they won't necessarily do that. They're still cancer, but they're not very, very aggressive and invasive like these are. And in prostate cancer, clinically, and in some of our mouse models, the cancers go to the bone and they stay there for a period of time. And we're really trying to understand why they go and stay in humans. We think for a period of time can be years. And then after a period of time, the cells that have been dormant, so to speak, or cellularly quiescent, they reawaken and are micrometastatic in the bone. And
Dr.
so,
Zarif,
yes.
We only have about 10 minutes, and I was wondering if you could answer a question that's appeared in the Q&A for us.
Yeah, this is I can stop here. Yep.
Sure. So Rachel Hudspeth is asking, can you share an experience from your career or in school when you were rejected from something that you really wanted, and how you handled that and overcame it to get to where you are today?
Yeah, so I applied to lots of examples. I'll just keep it scientific, but I applied to a few graduate schools. I did not hear back from one of them in particular until it was too late, but I heard back from Michigan State in time. So I wanted to go to Michigan State as well as this other place. I think I was on the second or third wait list or something, so they had to wait until a bunch of people said no to the school and then give me a call. But I was like, too late. I'm already going. It was like April or May or something when I heard back from them. So rejection can be a bit tough, but just try to look for a silver lining.
Can you share with us how many times you had to apply for grants before you were actually successful in landing them?
So the only grant that I did not get that I applied for was an R1. So I applied for NCI R1 last June. And because I hadn't published first on what we were studying, they wanted to see that. So that was really the only big rejection. Some of the other grants, the Challenge Award, the Young Investigator Award, I got on the first try. The K Award, I got on the first try. I had a pretty good lucky streak, I would say. But I had already published my findings, and so that substantiated, I guess, a reviewer's, desire to see that we had something in press already to substantiate some of the claims that we were making in the grant. So I haven't had a bad rejection streak, not yet at least, for getting grants.
Okay. If others have questions they want to post in the Q&A, please go ahead and do so. I've got another question. How do you approach mentoring for the individuals in your lab? How do you translate your experiences into the advice that you're giving people who are trying to make it in science?
Yeah, so I try to meet them where they are. I run my lab very differently than most people do. So I meet with every single person in my lab once a week for 30 minutes. Doesn't even sound hard, but you'd be surprised. Some mentors are so busy that they can't meet one-on-one with their trainees. They could bring an article, they could bring their current results, which most students do, most trainees do. Or we could talk about, I guess I don't really have issues in the lab, but if there was some, issue in the lab, we could also discuss that too. So one, I try to meet them where they are. I try to meet with them regularly, give them the encouragement that they need, and make sure that they don't have any hurdles in the way for some of the research studies that they want to do. Here, my job is to make sure students are successful, they don't have hurdles, at least, in the lab, hurdles in their way to do the work that they want to do, and to keep them, excited and want to come to the lab, having a well-resourced lab and things of that nature. My high school student, it was interesting because, he talked to me about different things than what, my postdoc would or someone else. He would say, Dr. Zarif, what do you think about this particular college? Or what do you, when he was on this scholarship hunt and things of that nature. And I tried to give him the advice that some of the best advice that I would give a 17-year-old version of myself or 16-year-old version of myself, and to keep an open mind, visit the campus, go with your parents, things of that nature. So that's how I handle.
Thank you for that. Can you describe what you think one of the biggest shocks was in transitioning from an undergraduate experience into graduate school?
For me, I went to a historically Black college, so for me I've never been the first Black anything until I went to Michigan State. I was the first African American student that the program had, I was the only African American student in almost all of my classes, so that was a shock a bit. But I didn't feel threatened or anything like that regarding it. So it was a shock in a good way. I would say some of the courses, just the amount of material that's given in an 800 level course versus it was a master's program, it was a 500 level course. It's pretty different. So you're responsible for a lot more of knowing a lot more than what you had to know for a master's or especially for undergrad. In my master's program there were no multiple choice tests. So I will say from my master's to undergrad, that was a shock there because there were no multiple choice tests like there were in undergrad. And the same was true at Michigan State. So I will go there. And then I will say finally for my master's, and this is what really helped prepare me for my PhD, was that the master's was really, really hard because you had to take all of these classes and do research at the same time, and you only had 2 years to do it. A PhD, you can spread your classes out, but the master's was so short, and you had to finish all your courses and your comps in a given time to make the deadlines. So that made it really tough. But I think those were the I guess growing pains with going into graduate school.
Thank you. And then the last question. In science, we like to sort of perpetrate a particular archetype of what a successful scientist looks like. And what that tends to do is marginalize elements of people's identity in order to conform into these archetypes. Can you tell me if you've struggled at all with that and how you've managed to hang on to particular aspects of your complex identity and use that to foster your own success?
Yeah, I guess the archetypes are like, some of, I guess, Black scientists come from, impoverished homes or whatever, or, I think everybody's story is unique, even if they came from, wealthy homes. I think that's still a unique story. I think you just, for me, I just followed what I was passionate about. And those really three things: the music, the science, and the martial arts. Everybody else got out the way. That's what I want to do. And I didn't let anybody tell me I couldn't do it. So that was me. Other people may have other passions that they're passionate about, or they accidentally found science and then they became passionate about it. But I think for me, at a very early age, like again, I wanted to understand how the record player works and things like that. It kept me interested in a basic science in a way. But I think everybody's story is unique. I just have mine. I think mine is just a bit moderate, more moderate than others that I've read about in Cell recently and things like that. But you're the, in a way, somewhat the author of your own story, and don't let where you come from determine where you can go. Don't let your past situations dictate what your future can be, no matter what those situations are.
Thank you so much. So we've come to the end of our time. We've posted the link for the student chat with Dr. Zarif that happens at 1:30 p.m. Houston time. So with that, thank you so much, Dr. Zarif, for the hospitality of your time. We greatly enjoyed your talk.
Thank you again for having me.
Thank you so much. Bye, everybody.